All right, I'm going to start talking about giving an update of our project, Epigenetic Landscape and Regulatory Divergence of Human Craniofacial Traits. I'll start from introducing the team members that are actually here at the meeting. We all know and love Lucia Celeri, but I also want to introduce a ridiculously talented graduate student of mine, Sarah Prescott, who is here at the meeting, and also Tomek Świgut, who is doing a lot of the data analysis for the project and will be communicating with the hub regarding the data transfer and analysis. So I think it would be preaching to the converted to say that cranial neural crest cells contribute to this fantastic variation of vertebrate craniofacial form. But in our case, we're really interested in emergence of human-specific traits and molecular underpinnings that underlie them. And when you think in the context of cranial neural crest, these structures are strong targets of selective pressure and are divergent between humans and our closest evolutionary relatives, chimpanzees. And these are not only uniquely human facial features, but also adaptation associated with the larger brain size, speech articulation, facial expressions, etc. And I also want to state another fairly obvious thing, I think, which is that it's now broadly accepted in evolutionary biology that cis-regulatory change is thought to underlie most of the morphological divergence between species, in particular between closely related species. So in some elegant studies in model organisms, it really became clear that cis-regular change, particularly in those distal regulatory elements called enhancers, play a very important role in directing morphological changes during evolution. So then it makes it imperative to identify enhancer regions that are uniquely human and are divergent between humans and other primates. So the question that we started with, can we systematically map cis-regulatory divergence in human and chimp neural crest cells? And this is where our aims for the FaceBase come in. So the first aim of our project is to characterize epigenetic landscapes and transcriptomes of human and chimpanzee neural crest cells, and to identify both conserved and species-specific cis-regulatory elements. So in other words, the goal is to provide reference maps from multiple human neural crest cells, genetic backgrounds, and multiple chimpanzees, and also provide some annotations to these maps such that will be fairly easy for the community to see if the elements are indeed uniquely human or if they are conserved throughout evolution. And in the second aim of our project, we propose to analyze activity of some of those really interesting candidate human-specific craniofacial enhancers in vivo using mouse model. So of course, early human or primate neural crest cells are not available for isolation in vivo. Therefore, we have developed several years back a protocol to derive cranial neural crest cells from human embryonic stem cells. And I'm happy to say that Ruchi Bajpai, who pioneered this work in my lab, is here with us today as well, and she's now an assistant professor at USC. And since then, we also learned how to maintain this population of post-migratory neural crest cells, and we can now, in fact, maintain them and expand these cells for 10 to 15 passages in undifferentiated state and without loss of gene expression patterns. And moreover, more recently, in my lab, Sarah Prescott extended this model to the chimpanzee, taking advantage of the chimpanzee IPS cells and developing a parallel model for differentiation of chimpanzee iPS cells to the neural crest cells. These chimp neural crest cells look exactly like human neural crest cells and have similar overall expression of characteristic neural crest markers, and we've done many validations to make sure that these cells are well staged, and we have a comparable human and chimpanzee cell population. And with those, now for the FaceBase project, we started generating comprehensive epigenomic maps of human and chimp cells. And what we've done so far, we have already performed lots of ChIP-seqs for transcription factors, including AP2 alpha and NR2F1, which we previously shown are important in establishing enhancer states in the crest cells. For general coactivator P300, for H3K4 monomethylation, H3K27 acetylation, H3K4 trimethylation, also H3K27 trimethylation, which I'm not showing you here. And even though we have not proposed it in the original application, the field is moving forward. Our colleague at Stanford developed a new technology called ATAC-seq, which is equivalent to the DNA-seq technology, allowing for mapping of hypersensitive sites, but with using much less cells and really streamlined protocol, and taking advantage of another enzyme called transposase. So we also incorporated this ATAC-seq protocol into our data, and we intend to make it available for the community. So we already generated the data for most of this ChIP-seq. So in fact, we ahead of the schedule with the game. And if you look at the snapshot, how the data looked like in Genome Browser from 3 different human genetic backgrounds and 2 different chimp genetic backgrounds, you immediately see that these epigenomic patterns are generally well conserved between human and chimp, but this is exactly what you would expect. And in this particular region that I'm showing you, there is overlap with 3 enhancers that had been previously tested in the Vista enhancer browser, or in Vista enhancer database that Axel is also involved in, and you can see all of this. All 3 have craniofacial expression. And if you take a genome-wide snapshot, you see that most of the mapped elements correspond to putative active enhancers. So we can map them with hypersensitivity, transcription factors, coactivators, and then use histone modifications to really assess the activity status. And if you do that, you can really quite well distinguish promoters from enhancers and active from poised elements. And the biggest category by far are active enhancers. Again, not unexpected, given what we now know in the field about gene regulation. And if you do annotations of these enhancers, they in fact show highly craniofacial ontologies. So this is good, but what actually is even better, we've done cross-referencing of our enhancers with a Vista database. So we took all elements that overlap with the 2,000 elements that have been already tested in Vista. And of course, in Vista, there's a subset of elements that are craniofacial, but in fact, probably most of their enhancers have activity in brain, heart, etc., etc. The database is meant at testing different expression patterns. And then we asked the enhancers that we're discovering, do they show preponderance for being active in particular cell types and tissue? And we were very pleased to say that, see that the most enriched was spatial mesenchyme, followed by the branchial arch cells. So we really think that we are capturing elements that are able to drive facial expression in vivo. So in terms of upcoming plans for release of some of these data, as I mentioned, we even ahead of schedule with the production, and we're thinking that by mid-2015, we can deposit all human ChIP-seq data sets from 3 different genetic backgrounds corresponding to all of these ChIP-seqs, and in addition also ATAC-seq data. So this will be 30, 40 libraries, and we'll probably want to deposit both simultaneously, both their raw sequencing data through the SRA repository and also work with the hub to deposit minimally processed ChIP-seq and ATAC-seq results for visualization in the FaceBase genome browser, such that anybody can just pull out the data and see how they look like and look at their favorite gene loci, etc. And of course, I think, again, will be important to deposit metadata sheets that have detailed description of how the data has been generated, what the quality control was, etc., so we can easily compare and communicate. And so in the next few slides, I just want to show you some of the initial interpretations or analysis of this data that we started to do. As I already mentioned, we're very much interested in evolutionary questions. So we were really interested in looking whether we can, in addition to this invariant enhancers that I already mentioned, detect species-biased enhancers that show either human or chimp bias. And you can imagine easily the sort of two major mechanisms by which they arise, which is either structural variation in the genome. So we have an enhancer here, but this region may be deleted in chimp or missing correspond to the structural variant, so have no clear orthology. And then another mechanism of variation, and in particular we were interested in, is a quantitative change at orthologous regions. So both regions are present in human and chimp, but there's a quantitative change, which in turn translates into quantitative change in epigenetic marks. So we indeed see some of the structural variation, and this is probably the minority of variation. But now I'm going to focus on the variation that results from the quantitative change. And to quantify it, we initially focused on H3K27 acetylation, because us and others in the past show that this mark is really highly correlated with enhancer activity state. And if you compare at those orthologous regions, H3K27 acetylation within two human or two chimp individuals, you see some variation associated either with experimental variation or genetic background, etc. That's relatively small. But if you compare chimp to human, then you see much higher level of variation, and you can start identifying or calling those species-biased enhancers either human-biased or chimp-biased that are much more divergent than those dictated just by the intra-species variation that we observe. And an important point is, even if we use K27 acetylation only to call these divergent regions, this is in fact predictive of biases in other genomic features. So what I'm showing you here is that we called human biased regions by K27 enrichment, and you see that they're biased in human and much lower enrichment in chimp, or we call the chimp biased regions. And then at this regions called by K27 differences, we're asking what are the differences in P300, ATAC-seq, and AP2 alpha, and as you can see here, the regions that are different for K27 acetylation are also differential for these other genomic or epigenomic features, suggesting that the K27 acetylation is a relatively downstream readout of enhancer activity state, and we can robustly use this for these calls. So just to give you a little sense how the data looks like, so here I'm showing the example of the chimp biased enhancer at the PAX3 locus. You go down the chromosome a little bit, you see human biased enhancers, and we can call also quantitative differences. So here's an example of a strong bias, and this is an example of the weakly biased enhancer. What you're looking at here are those P300, K27 acetyl K4 mono enrichments for a couple of individuals. And what the communities probably will be most interested to know is that, in fact, from this preliminary analysis, we can already say that the species-biased enhancers are, in fact, associated with genes that are known to affect craniofacial structures that are divergent in humans and chimps. And moreover, often we know that these genes can act in a dosage-dependent manners because we see multiple genes associated with human syndromes that are known to be caused by haploinsufficiencies, showing the chimp-biased or human-biased epigenomic landscape. So we think that this is really quite exciting, and if you perform functional annotations of these divergent enhancers, you really come up with quite interesting facial categories that will be interesting to explore in the future. And an important control is that these epigenomic predictions can be validated. In this case, we're using luciferase reporter assays. We're cloning the epigenomically predicted enhancers and the orthologous regions from human and chimp, and looking at either chimp-biased or human-biased elements. And the conclusion from is that not only the predictive regions or the regions predicted epigenomically are indeed enhancers, we also can validate the biases. So what we predict as chimp-biased is indeed chimp-biased in the luciferase assays, or what we predict as human-biased is human-biased. And this is true regardless whether we're performing our test in human or chimpanzee neural crest cells, telling us that the differences are encoded by sequence rather than determined by the differences in transregulatory environments. So this is all fine, but as you are developmental biologists, you probably cringe at the in vitro models. And while we think we've done very extensive validation of our in vitro model, it is important, of course, to ultimately take this in vivo. And this is where our second eye comes in. And Licia Celeri, who's a lead person on the second aim, is working with us to use transient transgenesis reporter assays in mice to test this human-biased candidate regions. And the idea here is that even the enhancer may be human-specific, nonetheless, it should be properly recognized by the trans-regulatory circuitry of the mouse, because trans-circuitry is much more conserved than cis-regulatory change that is driving the evolutionary change. So we have started doing this analysis, cloned the first set of constructs, and have right now ongoing analysis of the first 5 on the phase 5 construct, and 2 of which are complete thanks to the fact that Licia worked herself on this throughout the Christmas holidays. So I'm gonna show. I was getting those emails from her at midnight my time, which meant 3 a.m. New York time, and say, I see beautiful things. So I want to show you some of those beautiful things that Licia saw. But first, I want to introduce the region. We tested this differential strongly human-biased enhancers at the CNT-NAP2 locus, which is a gene associated with development of human speech. And indeed, when we test the human region in the mouse, we see expression in different domains in the head and face. And 5 of the 5 embryos show reproducible expression despite of the variation in intensity, which is commonly associated with this type of assays. And then when we compare this to the orthologous chimp regions, we see that human potentially, and I'll explain why, potentially gained this head expression domains. And within the head, we only see expression in the olfactory placoid for the chimp, but all the other domains, the midline groove, the midbrain hindbrain border, IPED, etc., appear to be human-specific. I have to qualify this because we only had 2 LAG-Z positive embryos for the chimp, so we unfortunately have to repeat the injection to get more numbers, but given the high reproducibility of these patterns, I'm quite optimistic. So that's our first constructs, and now the analysis of other reporters is ongoing, and the idea again is that by mid-2015 we can start putting in some of the first images to the database, and then, of course, clone and inject next round of constructs. And with this, I think my time is up. And I want to end by, again, thanking people that were involved, Licia, for the mouse transgenic analysis. Sarah was really a true driver of the chimp project. Tomek has done a lot of data analysis. I also thank Rusty and Carol from the SOLVE for sharing chimp iPS cells, and Robert helped with processing images. Thank you very much.
Any questions? Okay.
So in the regions where you see species-specific enhancer signals, do you see that there is more sequence divergence in those areas typically? I guess what I'm asking is, so is sort of sequence change at those locations the main driver, or is it the factor of binding?
It is. So obviously, I mean, it's not. We were quite surprised to see that those differentially regulated enhancers have so little sequence change. They typically have 3 to 6 base pair changes over the orthologous regions of 1 kb, which we think is actually quite exciting because it allows us to decipher what those specific sequence changes might mean. Nevertheless, we see the increase of divergence with the increase of the so-called Levenstein distance, which is a measure of the sequence, the number of mutations between the sequences, and that's definitely the case, that the more mutations you have, or the more sequence changes you have, the higher variance you observe.
But it's fairly limited.
But it's fairly limited, yes. Except, of course, those regions that are due to structural variation. Yeah. Yes.
So, I think you were saying that the peak size is, in some sense, predictive of the level of gene expression if you're just using that enhancer to drive the reporter.
Yeah.
Is it also predictive for expression of the endogenous gene?
And if you could repeat the question.
So if the peak size, in the reporter assays, the quantitative difference in the ChIP-seq peaks is then predictive of the quantitative difference in activity in luciferase reporter. So yes, that's the case. Is it true at endogenous loci? So we're still looking into the linking, doing the expression analysis and linking the changes in enhancer patterns to expression. Clearly, this is going to be much more complex, because enhancer landscapes are very complex, and quite often a single gene, particularly important craniofacial genes, would have 5 or 10 enhancers in vicinity. So deconvoluting those influences may be less than straightforward, obviously. Nevertheless, we think that at least we have some preliminary evidence suggesting that at least in some cases there is a corresponding effect on gene expression.
And there's one more, and we'll just take one more question.
I'm very excited about this data. There's one thing, in the human samples that we had multiple human lines.
Mm-hmm.
You find individual specific enhancers as well, and are these somehow correlated with why mine is not as dominant as the others, or cases out there?
So, obviously.
And if you could repeat.
Can we see intra-species variation? Essentially, we have multiple genetic backgrounds for humans. Can we now essentially identify the individual specific enhancers and then eventually correlate it with facial morphology? Yes, we probably could at some point, but it's a tricky proposition. First of all, I think to make any sort of distinguishing between the the potential noise of the experiment, we would probably have to have many more individuals and many more genetic backgrounds within species for testing, and maybe even ideally in familiar trios. So we don't have the data now. Nevertheless, on individual examples, yes, we can see examples where individuals individual humans differ in intensity, for example, of epigenomic marks. So clearly there will be variation there as well. But we focus just on what's there in all humans and not in the chimps, and vice versa, as the first pass.
I get that. So I was just thinking, extending the same idea a little more. Would you look at cohorts of human individuals who are what we call normal, what's the disease associated? Would you do prediction? There would be groups of enhancers that might be identified, and there's so many mutations. Predict these sets of enhancers, to screen in patients with X or Y or Z syndrome.
Yes, definitely.
Can you summarize the question?
Oh, so can we now, given that in a lot of GWAS studies and other studies, it's becoming clear that a lot of the disease-associated mutations are in fact within enhancers rather than protein coding sequences, that can we then predict some of those disease-associated enhancers and maybe incorporate them in some of the screening tools for clinicians? Again, it will be complex, but there are some cases for which the predictions would be very straightforward, meaning that we know that we see biases in enhancers at loci that are already known to be associated with haploinsufficient syndromes. Therefore, you may imagine that you can affect the dosage of the gene in multiple ways. One is through haploinsufficiency, the other is through changing the enhancer landscape. And in this case, let's say testing enhancers for such particular loci would make perfect sense.